This is the revision video for B3, proteins and enzymes. What are proteins and why are they so important? Proteins form many structures within our bodies. All proteins have a specific shape that enables them to carry out their function. Your cells would not function properly without these proteins. So where are proteins found? Which of the items above contain protein? The answer is all of them. Proteins are important structures in living things and are found in all the animal and plant produce shown above. We already know then that proteins are made within our cells from the code carried by our DNA. Each nucleus in a human cell contains 23 pairs of chromosomes. Those chromosomes are made from the chemical DNA, which forms a code to build those proteins. Short sections of DNA called genes contain the code for one particular protein to be made. A gene contains the code that enables a cell to link together amino acids into a long chain to make a specific protein. But not all cells need all the proteins that are coded for in your DNA. There are around 20,000 different genes within those 23 pairs of chromosomes, but a cell will only use the ones it needs to carry out its function. Genes that are needed by that cell will be switched on, while those it doesn't need are switched off, so that it does not make that particular protein. Different cells in your body have different jobs, so different cells will have different genes switched on. So why are proteins so important? What proteins are found in our bodies? Collagen is a structural protein needed to make skin, bone, cartilage, tendons, ligaments, and the walls of our blood vessels. The picture shows what collagen fibres look like. Another group of important proteins are hormones, chemical messengers that travel around our bodies in the bloodstream. An example is insulin, a hormone that regulates blood sugar levels. It is produced by the pancreas and travels around the body to the muscles and liver, where it regulates blood sugar by triggering the target cells to convert glucose into glycogen so it can be stored. People with type 1 diabetes do not make enough of their own insulin and have to inject themselves with insulin. Because insulin is a protein, it cannot be taken as an oral medication because it would be digested, just like the proteins in the food that we eat. Hemoglobin is another important protein that is needed to form red blood cells. Red blood cells contain the protein hemoglobin folded around 4 iron atoms. This protein is very important to carry oxygen around in the blood. Antibodies are also proteins. They are produced by white blood cells to destroy microbes that cause disease. On the outer surface of the cells in your body, there are receptors that detect signalling molecules like hormones and send messages to the inside of the cell. These cell surface receptors are also proteins. Also within cell membranes are channels that are made of protein that allow through large molecules that are too big to diffuse through the cell membrane. Another very important group of proteins are enzymes. Enzymes are biological catalysts. These are substances that speed up chemical reactions within living things. They speed up reactions such as photosynthesis, respiration, and protein synthesis. Like all proteins, enzymes have a particular shape that enables them to do their job. There is a particular area of an enzyme molecule called the active site. This is a very important part of the enzyme that the substrate molecule fits into. The substrate molecule is the reactant for the reaction that the enzyme catalyzes. Every enzyme only catalyzes one reaction because the active site is just the right shape for a particular substrate. Nothing else will fit. This is known as the lock and key hypothesis, as the enzyme and substrate fit together just like a key will only work in a certain lock. There are two ways an enzyme can work, either building large molecules from smaller ones, or breaking down a large molecule into smaller ones. The example above shows us smaller molecules being built up into larger ones. Firstly, the substrate molecules fit into the active site of the enzyme side by side. While the substrate molecules are in the active site, a bond is formed between them and one larger molecule is made. The product is then released from the active site. The enzyme is then free to catalyse another reaction between the same substrate molecules, because the enzyme is not actually used up in the reaction. The diagram shows us how all this sequence fits together to build a large molecule from smaller substrate molecules. This example shows us a larger molecule being split up into smaller ones. Firstly, the substrate molecule fits into the active site of the enzyme, and then the bond in the substrate molecule is broken. This is then released from the active site as two smaller product molecules. This type of reaction, where large molecules are broken down into smaller ones, is called a catabolic reaction. This diagram shows us how all this sequence fits together to break up a large substrate molecule into smaller molecules. Every enzyme in our body has a particular temperature and pH that it works its best at. This is called the optimum temperature and optimum pH. If the temperature is too low, the rate of reaction is low because the molecules do not have as much energy and therefore do not move around as much. If the temperature is too high or the pH is wrong, the shape of the active site is altered and so the substrate no longer fits, which then slows down the rate of reaction. When the shape of the active site is changed due to the wrong pH or the temperature being too high, we say that the enzyme has been denatured. This change is irreversible. The active site cannot go back to its original shape. A gene mutation is a change to a gene. If a gene is changed, this results in a different protein being made. Gene mutations involve a change in the sequence of base pairs in a section of DNA. The diagram above shows a deletion mutation, where one of the bases is removed. This results in a change in the sequence of bases. Mutations can also occur when an additional base is added in, or when a base is substituted for a different one. The altered sequence of bases results in different amino acids being joined together and a different protein being made. This protein may have a different shape and may not be able to do its job. But what causes these gene mutations? There are many things that can cause gene mutations. They can occur completely spontaneously with no external cause, often as a copying error when DNA is replicating itself. They can be caused by chemicals such as tar in tobacco smoke, or by exposure to ionising radiation such as X-rays and ultraviolet light. Not all mutations are harmful, but when they are, they can cause cells to divide uncontrollably, leading to cancer. They can cause a particular enzyme to not work properly, resulting in serious illness. They can cause different shaped protein molecules, such as the different shaped hemoglobin molecules seen in sickle cell anemia. Or they can cause malfunctioning protein channels, like in cystic fibrosis. Not all mutations are harmful though. Some mutations are neutral, meaning they are neither harmful nor do they offer an advantage. And some mutations can actually be useful. Evolution comes about due to mutations that offer the organism an advantage. An example of this is pale skin in humans, which came about due to a mutation. It is not advantageous to have pale skin if you live in a hot place, because the pale skin has less protection from strong sunlight. But for people who live in cooler climates, it allows their skin to make vitamin D when the weaker sunlight penetrates through their paler skin. But not everyone who lives in cooler climates has pale skin. Inuits have not evolved to have paler skin despite living in a cold climate. It is so cold where they live that their skin is covered so they are not exposed to the sunlight, and they already get plenty of vitamin D from all the fish in their diet.